Learning objectives include what is bactericidal drug and what is bacteriostatic drugs. And we'll also talk about some modes of action of some drugs with examples. Now, the word bactericidal means to kill. So those antibiotics or antibacterials or antimicrobials that kill the cell, they're called as bactericidal. And those that do not kill, they only arrest the growth of the microorganism, they are called bacteriostatic. This is an example of cell wall inhibition. Those drugs like penicillin, they target the cell wall. So when is the cell wall synthesized? It is synthesized when the cell is growing. So penicillin would target the microorganisms that are actively growing. So if the cell wall is not synthesized, the cell becomes weak, and it would result in the lysis of the cells. And so this penicillin is an example of a bactericidal drug. Some drugs inhibit protein synthesis. And as you know, the protein synthesis, when the protein synthesis is stopped, bacterial growth is stopped. But the cell is intact. So how come the bacteria then is not causing a problem there when it's right sitting there? Well, bacteria is stopped from growing by the drug in the body, and then the immune cells of the body takes over, and they then engulf that bacteria and kill them. So when the growth of the microorganism is stopped, their number is low. So body cells then can effectively clear them. And that is how bacteriostatic antibiotic work. So if that means if a person's immune system is not very strong, then bacteriostatic drugs would not work. So how specifically we can target the protein synthesis in the prokaryotes and sparing protein synthesis in the eukaryote is by looking at the structure of the ribosomes. We know that eukaryotic ribosome is different than prokaryotic ribosomes. So as you can see here, the ribosome has two components or two parts, 30S and 50S portion or parts. Various drugs, they bind to these ribosomes at different places or during different stages of protein synthesis, as we will see in the next few slides. For example, chlorophenicol. Chlorophenicol binds with the ribosomes in a way that it does not allow the peptide bond synthesis between the growing chain of the protein. So the protein gets stopped. Bacteria stop from multiplying, from growing. So these drugs that target proteins are essentially bacteriostatic. They have to be cleared by the body cells, by the immune cells of the body. Streptomycin is another example of a protein synthesis inhibitor. Streptomycin binds to the ribosomes where messenger RNA binds. So it binds in a way that the message from the messenger RNA is misread by the ribosome. So it inserts a wrong amino acid there. So when a wrong amino acid is inserted into the growing chain of that protein, it gets mutated. So a mutated protein is of no use. And that's how the bacteriostatic effect is seen. Tetracycline, on the other hand, also inhibits protein synthesis, but it interferes with the attachment of the transfer RNA to the messenger RNA. So this is another way an antibacterial or antibiotic targets protein synthesis inhibition. There are other drugs that target plasma membrane. For example, polypeptide antibiotic. They bring changes in the permeability of the membrane. So when the permeability of the membrane changes, cell can leak its content out into the environment, or water can enter into the cells. So the result is death of the cell. So these antibiotics, such antibiotics, must be bactericidal. Another example is an antifungal drug. fungi has sterols in their plasma membrane, and drugs like amphotericin B and ketoconazole, they interfere, they bind to sterol and interfere in the permeability of the plasma membrane. Other drugs bind to the nucleic acids so that replication is retarded. And the cells, once the DNA is not able to be duplicated, there would be no division, there would be no cell growth. But it is important to know that these antibacterials that target DNA, they could be sometimes toxic to the body as well. And a good example is, I think there's a sulfonamide, quinolones and rimpafim. These antibiotics target DNA. We'll see in another slide. But they could be toxic to the body. But remember that DNA targeting antimicrobials could be toxic to the body, and they have a very limited use. Still, other antimicrobials have a different target, like some can bind to the enzymes. Sulfonamides is a very good example of such an antibacterial. Sulfonamide bind to para-aminobenzoic acid. This PABA is a substrate for an enzyme that makes folic acid, and folic acid is needed for purine and pyrimidine synthesis. It's a part of DNA. So essentially DNA synthesis is inhibited. So in summary, there are various targets that are used by various antimicrobials. Some target the structural integrity, some DNA, some RNA, some protein, some plasma membrane.